So now we can say that adipocytes secrete lipoprotein lipases which go to the nearby capillaries and stick on the endothelial lining and keep on waiting whenever chylomicron passes by or VLDL passes by. They just bring out fatty acids and those fatty acids will go into this tissue and eventually they will go over there. So this is called lipoprotein lipases. Is that right? Now these lipoprotein lipases, right? Yes.
Do both the glycerols go back to?
Yeah, both glycerols go back. Now the question is that this is having fatty acids into it. What it has to do now? To synthesize the triglyceride, it should have glycerols as well. It should have glycerols as well. So what it is really doing? It has to synthesize its own glycerols. Now, how it will synthesize the glycerol? It can get the glucose in. It can get, these cells can get glucose in. When glucose comes in, they convert the glucose, this is glucose, into glycerol. And then there are enzymes which will take the fatty acids and glycerol together and produce triglyceride. Now you know how they store the energy. They store the energy in the form of triglycerides. But from where they get the raw material? To make the triglyceride, they must have glycerols and they must have fatty acids. How they get the fatty acids? These cells secrete actively lipoprotein lipases, which are expressed from the nearby endothelial cells, right? And from the chylomicron, which bring the exogenous triglycerides, and from the VLDL, which brings endogenous triglyceride. From there, lipoprotein lipases break away fatty acids and glycerol. Glycerols go back to the liver through the circulation, and fatty acids come into extracellular fluid, and then fatty acids are picked up by adipose cells. Meanwhile, adipose cells will also take up the glucose, and when it will take up the glucose, that glucose is used to synthesize glycerol, and then glycerol is fused with fatty acids, and eventually we have synthesized fat. Am I clear? Story is not so simple. Actually, this type of reaction should occur when you have eaten well. If you are very hungry, do you think that is the time this monkey starts storing the energy? No. It means this monkey behavior is different when you are in fed state. Fed state means you are very much nothing to do with the federal government. Fed state means that you have eaten a lot. And there's fasted state. Fasting state is when you have not eaten from many hours. Is that right? Now this monkey, adipocyte, should behave differently after the meal and should behave differently when you are in fasting state. I have just shown that this monkey is making triglyceride. So it means this stage will be when you have eaten a meal. It's very logical, isn't it? Now question is that, who tells this cell that we have eaten the meal and you should store the triglyceride? This is another question. Who tells adipocytes all over our body that we have eaten the meal and we should now start making triglyceride and storing energy? Answer is a hormone which all of you know. So who is going to tell me? Whenever you eat, when you have good meal, especially when you have carbohydrates and mixed meal with carbohydrate, proteins, some of the hormones in the body go up. One of the hormone just go to the fat cells and tell them happy news. There's a lot of meal and material about to come. So this fat cell immediately within few minutes, start synthesizing these lipoprotein lipases. It's just like that you get the news that someone will pass by the road with a lot of money and you ask your friend to stand there and manage it. You don't do like that because you are not fat cell. You have to understand the psyche of a fat cell. Now, what really happens that there is a hormone, attention please, there is a hormone which is a secret reporter and come to the fat cells and tell them, you see, something is coming good. Be ready to store the things. Which hormone signals fat cell, please start storing the energy, be ready for it. All of you know that hormone. What is that hormone? Yeah? Glucose is not a hormone. Insulin, that's great. Everyone should know insulin. Have you heard of insulin? So, insulin has so many functions. One of the function is, look, here's your GIT, here is your friend, pancreas. When more glucose is going, when you have eaten the food and glucose is going to the blood, what else comes from there? What is this? Insulin. What is it? Insulin. So after the meal, after good food, insulin level in the blood goes up. Insulin level goes high. Insulin has many functions. One of the function is it acts as a secret reporter to come to the fat cell and tell it that I'm here. It means a lot of energy is coming in the body. So you will be so happy to know there are special receptors for insulin on which cells? Fat cells. Right? So when insulin comes and fit here, right, the signals go to the fat cell. Signals go inside the fat cells. I will teach those signaling mechanisms in endocrinology, but you just trust me that insulin comes and stimulate a insulin receptor, and receptor gives multiple signals in the cell, and cell machinery become very active. First of all, what they do? They have special vesicles, and in those vesicles they have special type of, what are these? Channels. As soon as the signals come, these vesicles rush to the membrane, and this vesicle membrane and this membrane, they fuse with each other, and these channels get planted into cell membrane. What are these channels? These are channels for, these are transporters for glucose. So that it will take more and more glucose in and produce more and more glycerol. You understand it? That insulin has many functions. Insulin rush to the fat cells and stimulate the buzzer. And it start giving signals in the cell, and cell has already stored glucose transporters. And under the signal of insulin, activated insulin receptors, these glucose transporters are inserted in the cell membrane. And what happened? Glucose start trickling in, and now this glucose will be converted into glycerol. You will be surprised if glucose supply is too much. These cells are so clever that from the glucose they can directly synthesize fatty acids also. You see, they can take fatty acids from outside, or they can synthesize fatty acids with glucose. They have synthetic machinery and enzymes. So anyway, this is one thing these signals do. That these signals bring glucose channels to the surface. Another signal they do. You know what the signal they do? The signals go to the nucleus. Some of the signals go to the nucleus and activate a special genetic machinery. There is a gene present over here. One of the very special gene is activated. And that gene makes messenger RNA. And that messenger RNA is translated into a protein. And that protein, insulin came, stimulated the receptor signal to the nucleus, activate a gene. That gene is making messenger RNA. You know, DNA is transcribed into messenger RNA, and messenger RNA is translated into a protein, and that protein is thrown out, and this protein moves to the endothelial and stick to that. What this is doing? Insulin signals, insulin bind with the receptor activate the genes which are responsible to synthesize lipoprotein lipases. And messenger RNA is made. When messenger RNA is translated into protein, that protein is what? Lipoprotein lipase. Lipoprotein lipase is secreted by the cell and they rush to the endothelial cell and take their position so that whenever chylomicron come, they will bring the fatty acids out. You know, body is working so beautifully. Cells talk to each other. This is engineered by Master Nature. You get it? So actually, whenever you eat food, lot of insulin is released. It goes to the fat cells, and on the surface of the fat cells, it activates insulin receptors. Insulin receptors are activated. They give multiple signals in the fat cells. One signal will end up into more glucose transporters in the membrane, and more glucose will come into fat cell. Other signal will activate what? This will activate the genes, and those genes for the lipoprotein lipases, and cell will synthesize and secrete more lipoprotein lipases, so that nearby endothelial cells have lot of lipoprotein lipases that as soon as chylomicrons pass by, they will grab out the free fatty acids. Am I clear? Is it difficult to understand? This is how it works. So insulin is a very good signal that body is energy rich. Again, these cells are supposed to store energy. So they must get a signal when our body has extra energy. Our body has extra energy after the meal. So these cells will be responding to insulin. Now we come to the opposite situation. That what happens when you are very hungry? Let's suppose you don't get food for last maybe 15 hours. If you are not getting food from last long time, then what should happen? Then the mechanism should reverse. That's so simple. Somehow they should stop taking the glucose. Lipoprotein lipases should not work. Rather, when you are hungry, when you are fasting, these cells should release triglyceride energy. Actually, what happens? Number one, when you are hungry, when you have not eaten food, your blood glucose level goes down. So insulin levels go down. So when insulin levels go down, this stimulus is lost. Receptors are not activated. These signals are lost. Transporters are not there. Glucose uptake stop. Secondly, when there is reduced what? Insulin. When there is reduced glucose, there is reduced insulin. Receptors are less. These signals also stop. So genes for the lipoprotein lipases are not expressing, and concentration of lipoprotein lipases on the endothelial cell is reduced. Endothelial cell is reduced. So what we discussed up to now, that when person is fasting, blood has lower level of body fuel than energy. That is the time, number one, that is the time not to store the energy into fat cell. Number two, that is the time to release the energy from the fat cell. First of all, how not to store energy. Then I will explain how release energy. How you can explain that how these cells stop storing energy? Simply because when in the blood glucose level is down, it means insulin level is down. No more insulin stimulation, no more insulin receptor stimulation, no more availability of glucose transporter in the membrane, no more secretion, synthesis and secretion of lipoprotein lipases. Is that right? So energy storage stop. Second is that when you are very much hungry, man is an angry man. You know that or not? You know that, but it is true for women also, right? So hungry men are angry men, and angry men have high sympathetic activity or low sympathetic activity. High adrenaline and noradrenaline, or epinephrine or norepinephrine is more in the blood when you are hungry, or less in the blood? More. Remember, epinephrine or norepinephrine is released by adrenal gland. Adrenal gland releases lot of epinephrine, and sympathetic nerve ending release lot of norepinephrine. Epinephrine and norepinephrine are released in our body when we are under stress. When we are under stress. And of course, when you are very hungry, is it stress or not to your biological system?
Yes, stress.
That's a very bad stress. Try it for a few hours, right? Especially when your fasting state is more than 15 hours. You feel very stressed. You forget about many other things. You are looking for food, right? You are looking the food around in the world, and body is looking the food within itself. How? That when you are hungry, insulin levels go down and epinephrine and norepinephrine level go up. They are the signals to this cell to release energy. Insulin is energy storing signals. Epinephrine and norepinephrine are energy releasing signals. Is that right? So what really happens that in these cells there are special type of receptors. The special type of receptors, I didn't do it like this just for artistic reason. This is a receptor for epinephrine and norepinephrine, which has a peptide chain which 7 times passes through the membrane. We call them serpentine receptors. They look like snakes, right? These are the serpentine receptor for epinephrine and norepinephrine. So what really happens, let me tell you. When epinephrine and norepinephrine bind here, when you are very hungry, adrenal gland releases epinephrine and sympathetic nerve endings release norepinephrine, and epinephrine and norepinephrine does many functions in the body. One of the function is that this will go to the fat cells and stimulate the receptors, the special adrenergic receptors. We call it beta 3 adrenergic receptors. They are receptors for epinephrine and norepinephrine. They are stimulated. When these receptors are stimulated, they give signal in the cell. There is a special protein called G stimulatory protein. This G stimulatory protein is stimulated, and this stimulate another enzyme, and this enzyme is called adenylyl cyclase. And when adenylyl cyclase is activated, it breaks down the ATP into cyclic AMP. In the cell, cyclic AMP level will go up. Cyclic AMP level will go up. Again, it's so simple. Epinephrine, norepinephrine stimulate the receptor. Receptor is serpentine type receptors. All 7 pass receptors are coupled with G proteins. This is G stimulatory protein which stimulate adenylyl cyclase, which convert ATP into cyclic AMP. In the fat cell, cyclic AMP levels go up. When high levels of cyclic AMP drive an enzyme. What is the name of this enzyme? I will tell you the function of this enzyme, and then we'll talk about the name of this. This was our triglyceride. We have to break it, but we are starting whole strategy. Cyclic AMP fit into this enzyme, and when it is loaded with the enzyme becomes active. And when this enzyme becomes active, what it does? It will do something with another enzyme. Let me draw that enzyme, the other enzyme. This is an enzyme. Normally what happens, this enzyme exists in 2 forms. In one form, its mouth is closed. In another form, its mouth is open. In this form it is active, in that form it is inactive. So this enzyme works in 2 forms, inactive form and active form. Listen now carefully. Actually, the function of this enzyme is when it is activated, it breaks down what? And what it releases? Glycerol and fatty acids. This enzyme is present in the fat cells. This enzyme is normally present within the fat cells, but normally it is sleeping. This enzyme is inactive normally. Right? And here it is fully active. But how it gets from inactive state to active state? Actually, there is a point here. If you put here phosphate, what you bind here? Phosphate. Right? This phosphate will bind one point here, and other point it will bind here. So when it is having phosphate here, it converts into active form. When it does not have phosphate, it is inactive form. So you can say phosphate is the molecular crown for it. When it is phosphorylated, it starts its function. When it is dephosphorylated, it sleeps. Am I clear? So this enzyme right now is active or inactive? This is active. Why it is so active? Because it is phosphorylated. There is a crown on his head. And what is this crown? Phosphate. From where this phosphate come? Someone has to crown this enzyme. This is the function of this enzyme. Actually, cyclic AMP stimulates this enzyme, and this green enzyme take the ATP, takes ATP. ATP has how many phosphates? 3. So, it takes the ATP, break down into ADP, and take one phosphate and add to this. And when it is crowned, it becomes very happy, and it will start eating the triglyceride and start releasing fatty acid and glycerol. Is that right? Yeah? I can repeat. There is an enzyme, it's just like you. It's there. If it is not crowned, it is sleepy. As soon as it is crowned with phosphorylation, it becomes active. And when it becomes active, what it does? It catch up the nearby triglyceride molecules and clutch, breaking them down. And break them down into glycerol and fatty acids. And of course, fatty acids will go to the blood. This is a very important source of ready fuel. And glycerol will also go to the blood. This glycerol can go to the liver. And in the liver, this glycerol the carbons of glycerol can be converted into new glucose. So you can say this glycerol can be transported to the liver and converted into glucose, or can be converted into liver and another type of fuel molecule which is called ketone bodies. So glycerol can be used by the liver in 2 ways. For example, I'm very, very hungry. Then what will happen? In my body, glucose level goes down. When glucose level goes down, insulin goes down. Now fat cells don't pick up the glucose and they don't express lipoprotein lipases for the endothelial cells. So fat cells are no more cells. In the fasting state, they are no more energy storing cells. The behavior totally changes. Why? Because they are not under the direction of insulins. When you are hungry, fat cells are under the direction of epinephrine, norepinephrine, and some other hormones like that. Right? Insulin is energy storing hormone. We call it anabolic hormone. Epinephrine, norepinephrine, and their friends are catabolic hormone because they break down the fat. Am I clear? So when I'm very hungry, I'm very fasting, my body is producing more epinephrine and norepinephrine. It stimulates the fat cells, and what really happens, epinephrine and norepinephrine stimulates, it's a receptor, and receptor stimulates G stimulatory, which stimulates adenylyl cyclase, which convert ATP into cyclic AMP. So once epinephrine and norepinephrine stimulate the adipocyte inside the fat cells, which levels go up? Cyclic AMP. The cyclic AMP stimulates this enzyme. This enzyme is called, protein kinase A. Why this enzyme is called protein kinase A? Because it takes up the proteins and do kinase activity. Kinase activity is what? Phosphorylation. For example, I have this phosphate. And if I'm an enzyme, I take this phosphate, and if she is protein, if I put the phosphate on her head, I've done what function? Phosphorylation. This protein is phosphorylated. An enzyme should be called kinase. So whenever an enzyme phosphorylates other proteins, the enzymes are called kinases. Now, this is kinase, having kinase activity for target protein. This enzyme is protein. It is crowning them. So it is protein kinases enzyme, which are driven by AMP. So protein kinase A. Protein kinase A are an group of enzymes which are driven by cyclic AMP, and they can do phosphorylation of target proteins. Am I clear? So what we are doing, that adenylyl cyclase increases intracellular cyclic AMP, which increases protein kinase A activity, and they phosphorylate this enzyme. What is the name of this enzyme? This is also lipase. It breaks down the lipids. Now look, this is also lipase. It breaks down the triglyceride. This is also lipase. It is bringing up triglyceride. But these two lipases are different. What was the name of this lipase? Lipoprotein lipase. And this lipase is hormone sensitive lipase. It is sensitive to that hormone, epinephrine and norepinephrine. So what is the name of this enzyme? Hormone sensitive lipase. Now listen, my friends. Let's clear concepts. In the adipose tissue, hormone sensitive lipase are within the cell and lipoprotein lipases are on the endothelial cells. Again, in the adipose tissue consists of lot of fat cells and it is richly supplied with the blood and richly supplies with the nerves. Am I clear? In the adipose tissue, on the endothelial cell, we can find lipoprotein lipases. And within the adipose cell, we can find hormone sensitive lipases. Is that right? When we have eaten a meal, insulin level is high, epinephrine, norepinephrine level is low. When insulin level is high, that is a signal to the fat cells to store energy. So fat cells, when insulin levels are high, take up more glucose and they also produce more lipoprotein lipases so that triglyceride present in chylomicrons and VLDL can be catabolized and free fatty acids can be brought to this cell. And from the glucose, they can synthesize glycerol. And fatty acids can be made from the glucose or fatty acids can be extracted from the VLDL or can be extracted from the chylomicrons. Once fatty acids are present in the cell, right, they will be fused with the glycerol and produced triglyceride. And these triglycerides synthesis increases after a very good meal. Is that right? But once you don't get for many hours food, insulin level goes down. So anabolic signals go and anabolic activities go down. And when you are hungry for a long time, what is happening? Epinephrine, norepinephrine levels are high, and these levels which are high epinephrine and norepinephrine, they will stimulate beta-3 receptors, they will give intracellular signaling, stimulating G stimulatory proteins, stimulating adenylyl cyclase, increasing cellular cyclic AMP, protein kinase A are activated, which will phosphorylate hormone sensitive lipases, and activated hormone sensitive lipases break down the triglyceride into glycerol and fatty acids. Fatty acids will come into blood, they will act as a fuel in the body, and glycerol will be taken to the liver. There it can be used to make new glucose, which is called gluconeogenesis. Gluconeogenesis is a process in which glucose is made from non-carbohydrate sources. From the fat we are making glucose. You know, we've broken down the fat and we made glucose. This process is called gluconeogenesis. Glucogenesis means what? You are making glucose. Neo means from new sources. So gluconeogenesis. You are understanding? So this glycerol can be used in gluconeogenesis, and if there is extra glycerol, that can be converted into ketone bodies. And those ketone bodies also act as fuel. You will study in biochemistry. Is that right? Is there any question here? There is no question over here. Right. Now very briefly, I will talk about two types of obesities. Obesities, only just few words. Right, we'll discuss that in detail in biochemistry. Right now, obesity, of course, when you have a lot of extra fat in your body, there's two types of obesity. Number one is hypertrophic, other is called hypercellular. Listen, fat cells which are present at neonatal life, right? In early neonatal life, fat cells or adipocytes can multiply or proliferate. As your age increases, they lose the capacity to multiply. Now, if a very small infant is overfed, his fat cells will become small or large? Large. Plus they will multiply. So if a very small child, infant is overfed, his number of fat cells will increase as well as size of fat cells will increase and he will become obese. Is that right? But if there is an adult person, he overeats. Fat cells cannot increase their number because fat cells lose the capability to multiply with the adult age. But fat cells still have capacity to enlarge. Is that right? So what really happens in adult onset obesity, what really happens, the number of fat cells in your body are fixed, but you can really make them overloaded with triglyceride and you become yourself fat. This type of situation, when a small cell increases its size, is called hypertrophy. What is that called? Hypertrophy. So adult onset adult onset obesity is hypertrophic. Hypertrophic means small fat cell will convert into a large fat cell. But in early onset, child onset obesity, right, is basically there is a small fat cell. If you are overfeeding, not only this cell will first multiply into many cells, and eventually they will become very, very large. This is called hypercellular. The number of cell is increasing. Hypercellular obesity. This is childhood obesity, right? If mothers overfeed their children at very early stage, the number of fat cells will increase. And when you are having too much excessive number of fat cells, all your life you have trouble in controlling your weight. Fat cells number is also inherited. When we say some people have inherited tendency to be overweight, probably the genes which determine the number of fat cells are determining more fat cells number in your body. And if you inherited larger number of fat cells, right, at the top you eat more, it's very difficult to control your weight. Is that right? So your weight depends on so many factors. We'll discuss in detail in biochemistry. Here I'm just saying that obesity can be due to simply increase in the size of the fat cells. That is adult onset obesity. That is also called hypertrophic obesity. Or it may be early age onset obesity, and in which age fat cells can even multiply when they are overloaded with the fat, and we call it hyperplastic or hypercellular obesity. Hyperplasia means increase in number. So it can be called hypercellular obesity, or it can be called hyperplastic obesity. Right, and thank you very much, and lot of clapping for my son who is having a fever of 105 and still trying to shoot this lecture. Thank you very much.
Thank you.
Welcome. Yeah, he was